Welcome to Part-4 of Enzymology 4th unit. In the last class, in the previous class, we have been discussing about mechanism of enzyme action. In that, we have discussed two important mechanisms. First one is acid-base catalysis and second one is covalent catalysis. And third one is metal ion catalysis. In the metal ion catalysis, we have briefly I have briefly described about the metal ion catalysis, where some metal ions binds to the active site of the enzyme, and because of their positive charge they will stabilize the negative charge on the substrate and the transition state as well. So for the stabilization of substrate and transition state, the enzymes require positively charged metal ions for their activity. So thereby they will decrease the activation energy of reactants. They will orient the substrate in such a way the catalysis takes place precisely at the specific point. So orientation is also very important point when we consider the reduction in activation of energy. So if the substrate is oriented in a wrong direction, then the enzyme must use some energy, put some energy to reorient the substrate into appropriate position. So therefore, these metal ions play a key role in the orientation of substrates in the active site, thereby decreasing the activation energy. So that an enzyme effortlessly attacks at a specific site, and that requires very less energy for the reaction. So therefore, the metal ions will play a key role in the enzymatic catalytic processes. So some of the enzymes require positively charged ions like metal ions. Based on the metal ions bound to enzymes, the enzymes are also classified into two categories. First one is metalloenzyme. Metalloenzyme means the enzyme which bound a metal ion very tightly. So that metal ion cannot be removed from the enzyme. It is permanently bound to the enzyme. So those enzymes are called metalloenzymes. So we cannot dissociate the metal ion easily from the enzyme. So those kind of enzymes are metalloenzymes, and in these enzymes most of the metals used are ferrous ion, ferric ions, and copper, zinc, and manganese like metal ions. And next type of enzymes are the metal ion activated enzymes. So here the metal ion loosely binds enzyme. These metal ions include potassium, sodium, magnesium, and calcium. So these kind of metal ions loosely binds the enzymes and participate in the catalytic processes. So in metal ion catalysis, the role of metal ion is number one, binding to and orienting substrate for reaction. So substrate will be oriented in a proper way so that the catalysis takes place. Substrate binding and catalysis, two things are very important in the catalytic reaction. So substrate must be bound in a proper orientation. So that is assisted by the presence of metal ions, because the metal ions are having positive charges and substrates may have negative charges. So if they have negative charges, then the interaction takes place between the substrate negative charge and the metal ion positive charge, Therefore, the substrate will be oriented in a proper way. And the second one is they mediate redox reactions through changes in oxidation state. So they can also change the oxidation states. So that means suppose if you take the example of iron that exists in Fe plus 2 form and Fe plus 3 form also. Copper is also copper plus 1 and copper plus 2. So these kind of metal ions, they will change their reduction potential, and in the same way they can influence the redox potentials of substrates. So thereby they directly involve in the catalytic process. And third mechanism by metal ions is electrostatic stabilization and shielding of negative charges. So here we can observe the example in glycolysis. The hexokinase requires ATP along with magnesium ions. So for the transfer of phosphate from the ATP to glucose, it requires magnesium ions. So these magnesium ions binds the phosphate groups on the ATP and stabilize the ATP. They will shield the negative charge. So therefore, the phosphate group transfer takes place easily. So these are three major ways by which the metal ions will participate in the catalytic reaction mechanisms. And fourth and last mechanism of enzyme action is electrostatic interaction. Electrostatic means here each and every active site of any enzyme, they will bound a water molecule. They will bound water molecules in the absence of substrate. So the water molecule is already bound to the active site. Now the active site is not empty, it is occupied by water molecule. Once the substrate comes into contact with the active site of the enzyme, the water molecule will be released from the active site and substrate binds to that active site. So that substrate is replacing the water molecule. So once the water molecule comes out, what happens? The dielectric constant of active site will be decreased. So therefore, that substrate binds strongly to the active site. The interaction or will be the interaction between the substrate and the active site will be more. Therefore, that binding releases some binding energy. This binding energy is used as activation energy to drive the reactants to their transition state. So for the conversion of substrate to transition state, we require some amount of activation energy. So most part of the activation energy comes from the binding interaction between substrate and its active site, and it replaces the water molecule from the active site. So that binding energy will provide the necessary activation energy. So we do not need any external energy to activate the substrates or to convert the substrate to their transition state. So this is called electrostatic catalysis. So during the course of reactions, transition state is stabilized by electrostatic interaction. So the transition state, the intermediate state, here the enzyme substrate complex formation, that transition state will also be stabilized by the electrostatic interactions. So most of the enzymes will have amino acids which will have positive and negative charges. Because of their ionic charges, they interact with oppositely charged parts of the substrate and as well as transition state. So those interactions will stabilize the transition state in the active site. So that reduces the activation energy and increases the velocity of catalytic reaction. Okay. So far we have discussed about the four different mechanisms of enzyme action. First one is acid base catalysis, where some amino acid residues like histidine and acidic amino acids, basic amino acids, sulfhydryl containing amino acids, hydroxy group containing amino acids. These amino acids act as acids and as well as bases. They accept protons and they donate the protons also. So by this way, they convert the reactants to their transition states by acid base catalysis mechanism. And second one is covalent catalysis. In this mechanism, the amino acid residues will have positive charge or negative charge. They will develop positive or negative charge and they form a covalent bond with the substrate. So this bond, a covalent bond formation between substrate and amino acid residue in the active site of the enzyme will facilitate the formation of transition state. And the third one is metal ion catalysis. So all metal ions will are having positive charges. They bind the substrates and their transition states which are having negative charges, and they stabilize them. They will orient the substrates in a proper way also. They will shield the negative charges on the transition state from the outside environment. So thereby they increase the catalytic velocity and stabilize the transition state and reduce the activation energy required for the formation of transition state. And fourth one is electrostatic catalysis. In electrostatic catalysis, some amino acid residues which are having positive and negative charges, they electrostatically stabilize the transition states, and at the same time the binding energy between the substrate and active site, the active site of the enzyme, that will liberate some of the energy due to the formation of electrostatic interaction between the substrate ions and the charges on the residues in the active site of the enzyme. That energy utilized as the activation energy, so no further energy is required uh, from external source, and that substrate easily forms a transition state. So these are the four different mechanisms of enzyme action. And the next topic is regulation of enzyme activity. The enzyme activity can be regulated by three different mechanisms. First one is covalent modification, second one is zymogen activation, and third one is isoenzymes. Here the regulation of enzyme activity means the enzyme will be there, like in the case of allosteric enzymes, the enzyme will be in active state and inactive state. So based on the conditions, sometimes the enzyme must be switched off or inactivated, and sometimes the enzyme must be switched on or must be activated. Therefore these enzymes are regulated as per the needs of the cell. That is why enzymes will be regulated, and there are three different mechanisms by which these enzymes are regulated, and those three mechanisms are covalent modification, zymogen activation, and isoenzymes. Existence of isoenzymes. Here, the first one is covalent modification. Covalent modification is very important regulatory mechanism of enzymes. So most of the enzymes which are present in the cell by themselves, they will be in inactive form as long as they are not needed by the cell. So once a need is arised by the cell, then the inactive form of the enzyme will become active form. So the cell activates inactivated enzymes by covalent modification. In most of the cases, this is by phosphorylation. So a, if a phosphate group is attached to the enzyme, then the state of the enzyme will get changed. If it is dephosphorylated, again it's changed back to the original state. Okay. So that is called covalent modification. So the enzymes may be active or inactive in form. So they are interconvertible by means of covalent modification. So phosphorylation, dephosphorylation, and sometimes oxidation and reduction of disulfide bonds in the enzyme also plays a major role in the regulation of the enzyme activity. So the best example for this covalent modulation is glycogen phosphorylase. So glycogen phosphorylase is an enzyme which break glycogen into constituent glucose molecules, glucose 6 phosphate, glucose 1 phosphate molecules. So whenever the organism or individual requires cellular energy, or whenever the blood glucose levels decreases, what happens? The stored glycogen in the liver, they get they get split into glucose 6 glucose 1 phosphate by an enzyme named glycogen phosphorylase. So glycogen phosphorylase breaks the glycogen to form glucose 1 phosphate. Here the glycogen phosphorylase which is synthesized by the cell will be in inactive form. Whenever the cell requires glucose for the energy supply, then only the glycogen phosphorylase will be activated. So here when we look at the structure of glycogen phosphorylase, we will have a homodimer structure. So this is glycogen phosphorylase. We have two subunits, identical subunits, so that is why it is a homodimer. These two identical subunits will be in B form, form B, that is inactive form. So by the time they are synthesized, they will be in inactive form, that is in B form. So when a need arises for the glucose 1 phosphate, then a specific protein kinase known as cyclic AMP dependent protein kinase. So these protein kinases, in fact, they are activated by hormone mechanisms. Okay. So uh, the hormones will sense the lower glucose levels in the blood, and because of that the glucagon is an hormone that induces the breakdown of glycogen into glucose. So because of that hormone mechanism, cyclic AMP dependent protein kinases will be active. So these protein kinases will phosphorylate the glycogen phosphorylase which is in B form, that is inactive form. So because it is a kinase enzyme, it will attach phosphate molecules to the phosphorylase B. So this phosphorylated form is now known as phosphorylase A. So phosphorylase A is active form. This active form will break down the glycogen to glucose. Once the glucose levels are adequate, then what happens? A phosphatase enzyme comes and remove these phosphates so that the phosphorylase A becomes again phosphorylase B, which is inactive. So the activation and inactivation of the enzyme is through the phosphorylation and dephosphorylation by the corresponding enzymes. So this is known as covalent modulation. So most of the cases, most of the regulatory enzymes like a glycogen phosphorylase will be modulated by phosphorylation, dephosphorylation and oxidation and reduction mechanisms. Okay. Next one is zymogen activation. So here zymogen means the enzymes which are in inactive form. So these are, most of the zymogens are proteolytic enzymes in the digestive system. So in the digestive system, the hydrolytic enzymes which are synthesized, which are produced as inactive enzymes, the active counterparts of the digestive enzymes will be smaller than the inactive form of digestive enzymes. Here, most proteolytic enzymes are synthesized as larger inactive precursors known as proenzymes or zymogens. Sorry. These proenzymes undergo irreversible proteolytic cleavage to form active enzymes. So here, say for example, trypsin. Trypsin is synthesized by pancreas, and that trypsin en enters the duodenum, where it attacks on the proteins, polypeptide chains proteins in the food material or ingested material. So here this trypsin initially synthesized as trypsinogen or in the form of zymogen, that is known as trypsinogen or protrypsin. So suppose this is the trypsin and this is the active site of the trypsin. This active site of the trypsin will bind the protein molecules and split the peptide bond between specific amino acids. Okay. So when by the time of their synthesis, by the time of the synthesis of this trypsin enzyme, we will have a polypeptide chain that covers the active site. So therefore, this active site is no longer accessible for the substrate, that is the protein. So as long as this chain is covering the active site, this enzyme will be in zymogen form and it cannot catalyze the cleavage reaction. So for the activation of this enzyme, this zymogen or trypsinogen enzyme, we have to break the chain here. Then what happens? This chain will be removed, which is on the top of the active site. So now this proteolytic cleavage will expose the active site of the enzyme and the enzyme will be in active form. The enzyme becomes active and that catalyze the hydrolytic reactions. So these are called zymogens. So all enzymes, all proteolytic enzymes in the digestive system, they will be in zymogen form only, and so during their functioning, a proteolytic cleavage will convert the zymogen form to active enzyme form. So by the existence of these zymogen forms, the enzyme activity is regulated. Let us have this example, trypsinogen example, in which the enteropeptidase is the proteolytic enzyme, which is SRN protease, that will activate the trypsinogen to trypsin. So that will activate the trypsinogen by proteolytic cleavage. It cleave the sixth lysine residue, the sixth amino acid residue, that is lysine, the bond between the sixth amino acid and seventh amino acid peptide bond. Thereby, it removes a hexapeptide from the N terminal. So once the hexapeptide is lost from the trypsinogen, then the trypsinogen becomes trypsin, active trypsin. So here we can see, we can observe that this is the trypsinogen. In the trypsinogen, we have 1 to 6 amino acids. The sixth amino acid is lysine and seventh amino acid is isoleucine. Now the enteropeptidase, which is synthesized by duodenal mucosa, and it is a serine protease. This is also a serine protease, an enzyme, proteolytic enzyme. So this proteolytic enzyme specifically cleaves the bond between lysine and isoleucine. Okay, the peptide bond between sixth and seventh amino acid. So now what happens? This six numbered amino acid sequence, that is hexapeptide, is removed from the trypsinogen. So now from 7 to 245 is known as the trypsin, active trypsin. Once the trypsinogen becomes active trypsin, so it will be in active form. So it can the cleavage is irreversible. So the zymogen is irreversibly converted to active form. So here enteropeptidase, it is an activating enzyme which proteolytically cleaves the hexapeptide from the trypsinogen to make the trypsinogen a active enzyme, that is trypsin. And one more example is activation of chymotrypsin. In trypsin activation, the enteropeptidase plays a major role. Once a small amount of active trypsin is formed, that trypsin itself activate trypsinogen without the need of enteropeptidase. So instead of peptidase, the trypsin itself acts on the trypsinogen to make it active. So this is called autocatalysis. By autocatalysis, it can break its own hexapeptide, remove its own hexapeptide. So one active trypsin molecule can remove hexapeptide from inactive trypsinogen. Okay, instead of enteropeptidase, the active trypsin plays a major role here. So this is called autocatalysis. So this trypsin, the active form of trypsin, also involved in the mm, cleavage, proteolytic cleavage and activation of other enzymes like chymotrypsin. So chymotrypsin is like trypsin, it is also a proteolytic enzyme, and this proteolytic enzyme is synthesized as a precursor enzyme, that is chymotrypsinogen, and that is activated by trypsin and chymotrypsin itself. So when we look at the cat the catalytic activation mechanism of chymotrypsin, we have this number of 245 number of amino acids in a sequence, where the trypsin initially cleaves the peptide bond between 15 and 16. 15th amino acid is arginine and 16th amino acid is isoleucine. So trypsin breaks this bond. So we will get two fragments, one fragment with 15 amino acids and another fragments with remaining amount of amino acids. Then this chymotrypsin, intermediate form of chymotrypsin, that that becomes a substrate for active chymotrypsin. So that active chymotrypsin will remove two short peptide sequences. So one is from 14 to 15 amino acids, so that is a dipeptide having 14 and 15 amino acid, and another dipeptide having 147 and 148 amino acid. Two dipeptides will be removed from two different regions. So finally we will get a, a, a trifragment protein, and these three fragments are linked by means of disulfide bonds. So now it is the active chymotrypsin. So active chymotrypsin is having three different subunits or fragments linked by two different disulfide bonds. So here the trypsin is initiating the proteolytic cleavage process, and chymotrypsin uh, is completing the remaining catalytic process by autocatalysis, and then the chymotrypsinogen is converted to chymotrypsin, the active form. So this is known as regulation of enzyme activity by proteolytic cleavage. And final regulation mechanism is isoenzymes. Here isoenzyme means, iso means the same, enzyme means enzyme, same enzymes in terms of their activity. So different different enzymes with different structures, different physical properties, different ionic properties, different pH conditions, different Km values, different Vmax values, but both enzymes, all enzymes will carry out same chemical reaction. So those are called isoenzymes. For example, lactate dehydrogenase. So we have lactate dehydrogenase that convert pyruvate to lactate and lactate to pyruvate. So for this enzyme, we have more than one isoenzymes. Each isoenzyme will be present at different organs and carry out the same reaction at different conditions. So therefore they are all called as same enzymes, isoenzymes, but their amino acid sequence is different, and most of their kinetic behavior also different. So they are called isoenzymes. So they are similar only in terms of their reaction, and everything else is different. They are synthesized by different genes. They are called the isoenzymes. So one of the example is malate dehydrogenase, and that the malate dehydrogenase exists in two forms within each and every cell. The cytosolic malate dehydrogenase is completely different from mitochondrial malate dehydrogenase. So both are actually catalyzing the same reaction, both are known as same malate dehydrogenase, but they are completely different in their physical and kinetic behavior. They are called isoenzymes. Here the best example for the isoenzyme is lactate dehydrogenase. So for lactate dehydrogenase we have 5 different isoenzymes. They are named as LDH1, 2, 3, 4 and 5. They can be separated by electrophoresis. So because they are having different amino acid sequence, they will have different charges and net charges. Due to that reason, the electrophoretic mobility of these isoenzymes will be different. So therefore we can easily differentiate all 5 LDHs by using electrophoresis. When we look at the structure of LDH, the LDH is a tetrameric enzyme. The LDH will have four similar or dissimilar subunits, but those four subunits are two different types. So two different types are two different, and total number of subunits are four. So the first type is M subunit, that is basic and present in M stands for muscle and H stands for heart. So here when we look at all five LDH isoenzymes, the LDH1 will have H4 configuration. The constitution is H4. So that means the H type of subunits, four H type of subunits it will have, and it is present, it is found in heart and RBC. It will travel, the electrophoretic mobility is very fastest. It it will reach the bottom of gel, and LDH2 is having 1 M and 3 H3. LDH3 is having 2 M and 2 H2. LDH4 is having 3 M and 1 H, and LDH5 is having 4 M4 M 4 M's. So that is why we will find this LDH5 in skeletal muscle and liver. So based on the organ they are present, they will have different subunit composition. So when we study the significance of differential catalytic activity of LDH1 to 5, we will observe that LDH1, which is having H4 configuration, is predominantly found in heart muscle. Okay, so this favors the H4 subunits, or the LDH1 favors aerobic mechanism. This LDH1 is inhibited by pyruvate. The substrate is the pyruvate, and it is inhibited by pyruvate. Hence, the pyruvate is not converted to lactate in cardiac muscle, but it is converted to acetyl-CoA, which enters the citric acid cycle. So this LDH1 is inhibited by the pyruvate. So if pyruvate is there, then LDH1 is inhibited. So if LDH1 is inhibited, no lactate is formed, but instead the pyruvate is converted to acetyl-CoA that enters the citric acid cycle. So thereby it favors the aerobic respiration process in the heart. And when we look at uh, LDH5 M4, it is mostly present in skeletal muscle, and the inhibition of this enzyme by pyruvate is minimal. The pyruvate cannot easily inhibit the enzyme activity. So therefore the pyruvate is converted to lactate, and uh, uh, that that is that it favors the anaerobic respiration process. So that means in heart the aerobic respiration is favored, and in skeletal muscle anaerobic respiration is favored, and that is because of the different isoenzymes, isoform of this LDH1 and LDH5. And one more thing is the LDH5 has low Km, high affinity for the pyruvate, and LDH1 has high Km. So that is why the LDH5 in skeletal muscle quickly binds pyruvate and converted convert it to lactate. And due to high Km value of LDH1 for pyruvate, that pyruvate is not uh, converted to lactate because it inhibits LDH1 and favors the aerobic respiration process in which the pyruvate enters the citric acid cycle in the form of acetyl CoA. So this way, in different organisms, the same enzyme is having different isoforms, and each isoform is favoring the suitable conditions at their uh, sites. With this, we have finished discussing about the regulation of enzyme activity. Enzyme activity is regulated by covalent modulation, zymogen activation, and isoenzymes. And we have two important small subtopics in this fourth unit. First one is multi-enzyme complexes. Some of the enzymes, they are not simple enzymes, they are multi-enzyme complexes. That means they will have more than one activity and they can bind more than one substrates and carry out a complex reaction. So those kind of enzymes are called multi-enzyme complexes. And one particular example for this multi-enzyme complexes is PDH, phosphate dehydrogenase complex. So this phosphate dehydrogenase complex, which convert pyruvate to acetyl CoA. So it is not a single step. In fact, um, it involves three enzymatic reactions and five different coenzymes. Five different coenzymes are required and three different enzymatic activities are required in this reaction. Therefore, it has a complex. It is a complex, multi-enzyme complex with three different enzymatic activities. When we look at the enzyme activities of pyruvate dehydrogenase enzyme complex, we will have three enzymatic activities: E1, E2, E3. Pyruvate dehydrogenase, first one is, second one is dihydrolipoyl transacetylase, and third one is dihydrolipoyl dehydrogenase. And we will and it requires five different coenzymes like thymine pyrophosphate, lipomide, coenzyme A, FAD, and NAD plus. So these the PDH complex requires three different enzymatic activities and five different coenzymes to convert pyruvate to acetyl CoA. So these kind of enzymes with complex activities and more number of enzymatic processes uh, and more coenzymes requirement are known as multi-enzyme complexes. And the last topic, small topic, is the ribozyme. Ribozyme, the name itself explains that the enzymatic activity of RNA, ribonucleic acid, that is RNA. Some of the RNA molecules have shown their enzymatic activity. So those kind of RNA molecules which are showing enzymatic activity are known as ribozyme. The RNA molecules which are single stranded in nature, like polypeptide chains in proteins, these RNA chains can also interact with interact among them to form three-dimensional folding structures, like three-dimensional structure of proteins. Therefore, there are some examples which shows enzymatic activity and they obey Michael's-Menten equation also, Michael-Menten kinetics. So therefore, those RNA molecules are regarded as enzymes, but they are not proteins. Thereby, they are called as ribozymes. So one of the example, the first discovered ribozyme is ribonuclease P. This enzyme will cleave tRNA, precursor tRNA to functional tRNA. So, when the scientists that Altman and his co-workers identified ribonuclease P, they tried to study that ribonuclease P. They found a RNA component in that enzyme. That RNA component is cleaving the precursor tRNA. So therefore, they found that RNA component is responsible for the cleavage of precursor tRNA. So that means the RNA itself acting as a nucleo nuclease enzyme, acting as nuclease enzyme and converting the precursor tRNA to tRNA, and it is obeying the Michaelis-Menten kinetics. Therefore, this RNA, the ribonuclease P, is regarded as a ribozyme. So far in this fourth unit, we have been discussed about the factors which affect the enzyme catalysis, like substrate concentration, enzyme concentration, temperature, and pH. And after that, we have discussed about enzyme regulation, sorry, enzyme inhibition. So in that we have three different topics. First topic is reversible inhibition, second one is irreversible inhibition, third one is allosteric regulation. So in reversible inhibition, that is subdivided into um, competitive and non-competitive. Irreversible is completely irreversible, and they are poisonous and toxic substances, and we have studied some of the examples also, like iodoestate, diisopropyl fluorophosphate, and organophosphorus insecticides. And the next important topic we have discussed is allosteric regulation, and we studied one specific example, that is ATC, acetyl transcarbamylase ATCase enzyme, which is the key regulatory enzyme, first enzyme of the pyrimidine biosynthesis pathway. And we have discussed about the regulation process, what are the activators and what are the inhibitors, and how they activate and inhibit the ATCase according to the cellular needs. And then we started discussing about the regulation mechanism of action. In that, we have discussed about acid-base catalysis, covalent catalysis, electrostatic catalysis, and metal ion catalysis. Four different mechanisms we have discussed. And finally, we have seen the regulation process of enzymes. First one is covalent modulation, second one is zymogen activation, third one is isoenzymes. In the covalent modification, phosphorylation, dephosphorylation of glycose and phosphorylase enzyme, and in zymogen activation we have discussed about trypsin and chymotrypsin. And in case of isoenzymes, the LDH we have taken as an example, where we have LDH 1 to 5 different LDH isoenzymes. And finally, two small topics, like multi-enzyme complexes, in which we have discussed about pyruvate dehydrogenase complex, and the last topic is ribozyme, that RNA molecule sometimes acts as only there are two or three examples are there, not all RNA molecules, just two or three examples are there where the RNA molecule have shown to have the catalytic activity. So therefore they are called as ribozymes. So hence when we'll study the structure of, when we talk about the structure of a, an enzyme, we cannot strictly say that enzyme is a protein, because the ribozyme is the exception. Except ribozyme, all enzymes are proteins. Okay, so this is about the fourth unit, and thank you.